Next up, we have Dr. Deepa Agashe. She is an intermediate fellow at NCBS Bengaluru. Deepa is studying how restoring ancestral microbiome aids insect adaptation to new diets. So over to you, Deepa.
Okay, thanks everybody for being here still. I am happy to tell you about some of the work that we've been doing. This was a little bit of a slightly boring slide, as I realized after reading Sarah's email in a bit more detail, that this is meant to be for a general audience. So let me revise my starting slide. The gist of what I'm going to tell you about today is that old friends can make new shores possible and plausible, right? And hopefully it will become clear what I mean by this in a little bit. It's also a nice opportunity to show you this beautiful picture that my student Kritika took as part of this science photography workshop at NCBS. And I really like the imagination that went into making this picture one of my favorites. So hopefully again, I'll tell you more about this picture a little bit later. So what do I mean by old friends making new places tenable? I'm talking about the microbiome here, which I think all of you have heard about and probably read newspaper reports about all the time. As you probably know, the microbiome is referring to the collection of microbes that inhabit pretty much every part of our body, inside, around our body, and so on. And every little niche of our body seems to have its own unique microbiome, including bacteria, fungi, protists, right? They all seem to play a role in regulating our development, our behavior, our evolution. And we're interested in asking how these processes occur in general and how they evolve in general. So just to give you some context for this, about 1.5 kilos of a healthy adult human is expected to be microbial cells. So if you have antibiotics ever, you should expect to lose some weight. It's a good thing. And there are over 10,000 types of bacteria alone in our bodies, and about half the cells of our bodies are thought to consist, are estimated to consist of bacteria or other microbes, right? So that's an enormous proportion of microbes that we're really carrying around with us, and we really want to understand, well, how does this whole situation, how does this interaction occur, how is it established and maintained? And just to quickly give you some more background on why these microbes matter, I'm sure you've heard of studies with mice showing that the degree of obesity or the degree of fat that you put on as body weight depends on this weird, intricate interaction between your genotype, your diet, and the microbes that you have in your body, the gut microbes that help you digest your food. And then here's this one of my favorite examples of what's happening in the insect world with microbes. So these insects you see here on the left, this sort of darker insect, which is, it's a beetle. It is what a normal insect should look like, this particular species. And the darkness of the cuticle reflects the fact that it is properly sclerotized and is very tough and hard, which makes it hard for predators to catch it or for this beetle to be sort of crushed and squished easily, right, when it's moving around. And then on the right, you see a beetle which has been grown up in azanic conditions, so no microbes are allowed. And the reason this happens then is this beetle depends on this one specific species of bacteria, which it houses in specific pouches, okay, along its gut. And then this bacterium produces a precursor metabolite, which is critical for the beetle to sclerotize its cuticle, right? So without this precursor molecule, the beetle doesn't have enough of whatever is required to make the cuticle hard enough. And what's really fascinating about this story is these bacteria are recycled by the beetle when it's done using them, right? So every generation, when the beetle is now an adult, its cuticle is formed, it really no longer needs the bacteria. So it recycles them, there's autophagy that occurs, that nice pouch which the beetle has produced is reabsorbed into the beetle gut, and just a few bacteria are sent off to be attached to the ovaries so that they can be transmitted to the next generation when the female lays eggs, right? So, I mean, there's really, really interesting details to be had in the story. And so in general, the picture that has emerged from the last a couple decades of work is that host bacterial associations are intricate and they're pervasive, they're everywhere. And I want to just quickly touch upon a couple really interesting contrasting examples to this that we discovered in our lab in the last few years. One is that butterflies, which you see eating different kinds of plants and drinking different kinds of nectar, they seem to have evolved all these different diets that they use without relying on bacteria, which is a very contrasting pattern to what people observe in general, right? So most insects, hundreds of insects which people have studied for microbial associations, have at least some reliance on microbe, right? Whereas this is one case where a lot of species of butterflies, all the species of butterflies we've studied so far, don't seem to really care about the microbes they have. Similarly, dragonflies, which we sampled from different parts of India, also do not seem to really care about the microbes. They seem to have a transient gut flora that is really just reflecting what they ate in that particular season, in that particular location. So given these two contrasting examples, I've been thinking a lot about, well, what makes most insects have these established microbial relationships, whereas some insects seem to really not follow this pattern. And to get at this, one important factor that we must keep in mind is actually these kinds of relationships are surprising. Why are they surprising? There's so much change happening all around us, right? I mean, as we grow up, we eat different foods, our health changes, our lifestyle changes. Well, how do our microbes still deal with us then, given that we change so many things just in terms of what food we're providing them? So just as a quick example, here are 3 different conditions, places where we expect to find microbes. We know of really nice interactions between plants and microbes, but as a plant grows, germinates, there are many, many changes that occur in the soil around it. The plant is exuding different metabolites, allowing different carbon sources to be available to the bacteria and so on. And so the bacteria that are associated with these plants have to deal with that fact. Similarly here, you see a case where, I don't know why this video has stopped, but anyway, you see a case where these are ladybug eggs that are hatching, right? And again, during the process of hatching, the microbial community changes, the ladybug larvae come out, they first cannibalize on other eggs that siblings haven't hatched yet. They go on to then eat aphids as their primary diet. Eventually, they will emerge into ladybugs and do other things. And finally, this bottom video here, it's a time lapse of the carbon dioxide emissions at a global scale, right, that occur across a year. And again, so this is South America, Africa, and India. And you see that these are huge changes that occur even at this very, very major factor that alters what kinds of global carbon cycles happen, right? So all of these changes should make it very hard to establish a relationship between a host and a microbe and to maintain that relationship over time. So it is surprising that it occurs so often, and so we want to understand what allows such relationships to be established and maintained over time. And just as a quick example, I'm sure if you think back to many relationships you have had over your life as friends, with partners, whatever, it's hard work to maintain these things, right? So we want to know what happens. Okay, so going back to the questions that I wanted to ask, in terms of thinking about how, despite such dynamic changes that occur across organisms' lifetimes, how do these relationships get maintained? We wanted to specifically look at the example of host dietary shifts, which are very, very common, especially in the insect world. And we wanted to use these flower beetles as our model organisms. So this is the red flower beetle. Many of you have seen this in your homes, perhaps. It infests cereal flowers, most typically wheat flour. And beetles are best adapted to wheat, but they can adapt to new resources very, very rapidly in the lab. So I did some of this work in my PhD, where I had beetle populations adapting and evolving to use corn, for example, which is typically a really poor resource for them, but they can adapt in a PhD lifetime, right? So it is very fast. And so we use this system then to understand, as the host diet shifts, what happens to the microbiome? Is there a dependence? How does the relationship change? So to start understanding this, what we've done is we first took flower beetles, had them grow up in different kinds of resources, where wheat is the ancestral resource, and then corn and sorghum or finger millet are alternative resources that they could be using, but are really bad at. And we extracted DNA from the whole beetle and sequenced and amplified just a small piece, which is bacteria specific, the 16S ribosomal RNA part of that. And we use that to infer or to understand which kinds of bacteria are present in these beetles and in what proportions. So here are some data showing you what we see. On the y-axis is the relative abundance of different kinds of bacteria. Here I've grouped them at a very large taxonomic level, so we're thinking of phyla, not specific species or genera. The green are firmicutes, and the yellow are proteobacteria, right? And these are just groups that are composed of thousands of species themselves. So what you see very clearly is that in wheat, in the normal wheat beetle, most of the bacteria are proteobacteria, right? And there are, the second dominant group is firmicutes. There are a whole bunch of others that I'm not going to worry about at the moment. If you now put beetles in these 3 different new resources, finger millet, corn or sorghum, you see a very different pattern. In general, sorry, I forgot to mention, each column you see here is one individual beetle. So there's some variation across individuals as well. So what you see here as a big pattern is that there are more firmicutes in all these new resources compared to proteobacteria, and so that proportion seems to be disturbed when you take beetles and you introduce them to new resources for just one generation. So this is like if I suddenly ask you to eat only soybean for a while, something is going to change in your gut microbiome, right? That's what we're observing here. So now the question is, do these beetles really care about the fact that the bacteria are different when they move to new resources? And so to test this, we took again individual female beetles. We either gave them flour, which is normal flour, wheat, corn, whatever, or we treated the flour with UV irradiation to reduce the bacterial loads in the flour. And then we asked the females to lay eggs, and we count how many eggs the females have laid. What we see is as follows. So number of eggs per female is on the y-axis. Here is the wheat situation, where these are control individuals that are laying eggs in normal flour, and they are laying about 20 odd eggs on average. And interestingly now, when you give them UV treated flour which has lower bacterial loads, you find that their egg production or their egg laying is reduced by about half. So that's a dramatic difference. But interestingly, in the other new resources, even though there are overall changes in fecundity, we don't see that the beetles care about microbes, right? So in these new resources where they're not adapted, it really doesn't seem to matter that there are microbes or there are no microbes. And remember, I showed you earlier that the microbes here are quite different from the microbes in wheat overall, right? So what happens then when these beetles shift and are adapting to new environments, new resources? Sorry, I forgot to mention that we see similar results with antibiotics. So just to make sure that this UV treatment is not something odd and funny, we gave treatments with antibiotics, broad spectrum antibiotics. We see very similar patterns. We also find that other aspects of beetle fitness are changing. So beetle survival, lifespan, body mass, development rate, many different aspects are altered, right? So it seems like the beetles have evolved this relationship with some microbes, some bacteria in their guts, which confers some benefit. Okay? So now what happens when we allow them to evolve? So here what I'm showing you is just a schematic for how we did this experiment in the lab. Simply we take some beetles, some females and males, put them in fresh flour, allow them to lay eggs, the progeny develop, we discard the old flour, and we separate the adults which start the next generation. So one cycle is one generation, and we continue this process for many generations in the lab. Like I said, this is within a PhD lifespan situation. So we see within about 25-30 generations, which is on the x-axis here, the number of adults, which is on the y-axis, changes a lot, right? So you start off with whatever 200, 500 adults. There's a crash in the beginning, and then some populations shown in black here manage to adapt over time and have a positive growth rate where the number of adults is increasing with time, whereas some others called nada are red, shown in red here, and they barely survive. They don't go extinct. There are a whole number of other populations that manage to go extinct, and so they are out of this equation. But there are some that sort of scrape by, they're hanging in there. There are some adults, they're not quite extinct, but they're not showing a positive growth rate. So these two class of populations now serve as comparisons for us to ask, well, what is going on with the microbiome in these populations? So we did this egg fecundity assay again. Again, the y-axis is number of eggs per female. On the left side here, you see there are control females which laid eggs. This is the ancestor, laying eggs in control sorghum versus UV treated sorghum. There's no difference, I showed you this earlier. And all of the evolved populations, if we take females from these and we now give them flour which is UV treated versus not, we see a big difference, right? So we're recovering this old phenotype which was seen only in wheat flour, that if you have wheat microbes, you care about it, any other microbes you don't. So this suggested that something has changed in the microbiome of these evolved populations, right? Because in normal sorghum flour, they didn't seem to care earlier. Okay, so what has changed? Again, we did the same kind of 16S amplicon sequencing analysis I told you about before. These are just the same data I showed you earlier. In wheat, you have mostly proteobacteria, a few firmicutes. In ancestral sorghum flour, there are more firmicutes, a few proteobacteria. And now if you look at the 3 different evolved populations, again each column is an individual beetle, you find that these patterns look a lot more like the ancestral wheat patterns, right? So there are more proteobacteria and fewer firmicutes in the evolved sorghum populations. And if you look at the populations which did not adapt to sorghum, they retain the signature of many more firmicutes and fewer proteobacteria, right? So it seems that over time somehow these microbiomes have shifted to look like wheat microbiome again, even though in the beginning I showed you in one generation there's a shift away from the wheat microbiome. So this begs the question, well, functionally are these microbes similar or not to what we started off with? And so to test this, we took UV treated flour and introduced larvae from these sorghum adapted populations, which should then put their fecal matter, including the new microbes, in that flour, right? And so now we can ask, at a function level, if these microbes are introduced in that flour, what do the females think of that flour? We find that we recapitulate that old result where if we put these evolved microbes even in wheat flour or in sorghum, naive females which have never seen sorghum before, again seem to care about these microbes, and they lay more eggs even when we give them the new evolved microbes, not the ancestral wheat microbes, right? And so this suggests that at a function level, the evolved bacteria behave like wheat bacteria. And so to summarize, this is a cartoon of what we think is happening. You start with wheat beetles that have some normal microbiome dominated by 2 groups of bacteria. You put them in sorghum flour and their numbers drop. Simultaneously, their microbiome is imbalanced, it's different. Over the course of this evolutionary cycle, this rebalance seems to be restored. Remember that these beetles live in the flour, and so their fecal matter is continuously cycling through the flour, which is why we think these communities change with time. And eventually, we're back to a community that looks very similar to the starting wheat microbiome. At the same time, the beetles are increasing in number, right? So this is just setting the stage for what we really want to do now. This is about a year or 2 of work on this grant. And now we can ask the really interesting questions, which is which bacteria are critical and why. I'm sure many of you are going to ask me that or thinking about it. What is the impact of host association on the bacterial evolution itself? And why do some species have strong microbial associations but others do not? Right? I'd like to be at that last question in a few years. Okay, so I'd like to end by thanking my funding sources and my wonderful team of people in my lab who have helped to do this work in one way or another. This is Aparna, the PhD student who has done most of the beetle work I told you about before. And Kritika is another PhD student who did the work with butterflies that I mentioned earlier. I'm happy to take questions if there's time. Thanks.
We're running late, so again, Deepa, I hope it's okay if people catch you at your poster. Thank you once again for a really, really nice talk.